This protocol utilizes a widely used technique along with reagents and tools that can be acquired commercially to assess endogenous IR5 activation during the early events of stimulation. It can easily be applied for probing IR5 biology in other cellular contexts.
While native page is a widely used technique, obtaining clear, robust, interpretable, and reproducible results can be technically challenging. Using a commercial running buffer and gel system helps to minimize variability, and paying attention to details is essential, and experience is key to success in obtaining good results.
This modified native page protocol involves many steps with subtle details that are key to success. Visual demonstration can show these details that would be beneficial for researchers.
Maintain Cal-1 cell culture in a T75 flask at 37 degrees Celsius and 5% carbon dioxide with complete RPMI 1640 medium. When ready to stimulate the cells, transfer them to a 50 milliliter conical tube and centrifuge them at 200 times G for 5 minutes. Remove the supernatant and resuspend the pellet in medium to obtain a homogeneous single-cell suspension. Then count the cells with a hemocytometer and seed them at a density of 1 million cells per well in a 6-well plate with 4 milliliters of preheated medium in each well. Incubate the cells for 20 to 24 hours to allow the confluency to reach 90 to 95%. The next day, stimulate the cells by adding 4 microliters of 1 milligram per milliliter.